So today we're going to talk about with you about why do we need human tissue reference lipidomes, and to show some example from our study on the lipidomic characterization of human white adipose tissue. So we all know very well that lipids are widely used as a very useful readout of human metabolism. And you probably all know this cartoon from the biochemistry books, where we have the distribution of lipids in our body via different types of lipoproteins, and lipids which are coming in from the food intake are distributed and shuffled around in the different forms. And we also know that the clinical panels exist, which allows patients to be assessed based on their lipid readout and to make some conclusion about their health. So that is usually total lipids, serum total cholesterol, HDL cholesterol, triacylglycerides, serum phospholipids, and so on. But realistically speaking, what we actually now measure in the clinical practice is nothing more than this kind of cartoon cycles, which are shown on this diagram as well. Yeah, so we can say there are more or less cholesterol, so more or less this orange part, or more yellow triacylglyceride, and so on. So the question now, if we are capable to step further and actually have a real look what is going on, not just showing this kind of cartoons. And indeed, the development of mass spectrometry, and our group is really focused on the application of mass spectrometry-based lipidomics, provide us access to the identity of lipid species and lipid molecular species in complex biological samples. So using liquid chromatography, mass spectrometry, or some people use it without liquid chromatography, we can actually identify lipids, for instance, on the lipid species level, as it's shown here, or in many cases nowadays in routine application, we can do it even on the lipid molecular species level. We can also perform quantification and compare different conditions. So again, disease versus healthy conditions, and we do it in the relative manner. So we can say, okay, there are twofold increase of a certain lipid in disease. Or we can also do it with more effort on the absolute quantification level and actually report the concentrations of the lipids in the biological samples in disease and healthy conditions. So the majority of the studies which we and others are doing in this respect, in this very high throughput manner, report us something like that. So we can say that there is a lower level of lipid X, and this is associated with the condition I. So we can always plot that kind of graphs and say, okay, there is maybe a biomarker for the certain conditions. And that, of course, a very useful approach, because it allows to screen very fast large cohorts of samples from the patient, and we're really getting some insights. But what the question which I ask myself after we get such kind of data is something like that. So, okay, I can see the difference, but what it actually means. And if we want to make another step forward, we have to go from this observation of correlation biomarkers to more systems-wide mechanistic understanding of what is actually happening in certain pathology in the human metabolism. Yeah, so we have to be able to map these changes which occurs in this complex lipidome to the maps of the metabolic pathways and connections which are present there. So what we know that lipids, of course, these lipid signatures can report us some mechanistic understanding. And with development of lipidomics technology and in general, the lipids gained a lot of popularity in the last years. We know that this classical separation of lipids into signaling, energy metabolism lipids, or membrane lipids is already not so straightforward and becomes more blur these borders, so lipids actually plays different roles in different conditions. And that is connected with, I would say, kind of unique feature of lipids, that they do not usually act as a single molecule in most of the situations, but they actually usually have a cooperative actions. And these lipid collectives, in one way or another, serve as a functional unit in the lipidome. And this is, of course, very easy to imagine on the level of the membranes. Yeah, so when we have certain membranes, the single lipid in the membrane will hardly describe its property, but we have to look on this lipid collective to understand how the biological membrane function and what happens to it. But it's also true on the level of metabolism. So you could see here a very general representation of the overall lipid metabolic reactions, and clearly they are all very interconnected, and changes in one of the branch will actually lead to the changes in the other. For instance, accumulation of free fatty acids will result in increased shuttling to beta oxidation, and when cell cannot manage that anymore efficiently, we will start to accumulate and store safely lipids and lipid droplets. But if that is not happening efficient enough, we start to see some other, let's say, lipotoxic effects with accumulation of ceramides and so on. So all these reactions are very closely connected between each other, and changes in certain pathways will result in changes of the others, or there are also some blocks which might be much more resistant, and this is also interesting to know why certain metabolic reactions are not affected by changing of the overall lipid metabolism. So with this in mind, actually, it brings us to the point of the understanding what happens on the molecular level in lipid metabolism. And the question is, how much do we actually know about lipidomes of different human tissues? Can we address the specificity of this lipidomes remodeling in certain pathological conditions at all? And more interestingly, in age, gender, ethnicity dependent manner, yeah, because we know that lipidomic signatures, for instance, all in male, females, they differ, and that must mean something. And with that in mind, can we actually use blood plasma lipidome as a readout for a tissue dysfunction? So for that, to address this question, we need to describe this tissue reference lipidomes as a qualitative and quantitative inventory of lipid molecular species, in particular tissue in organ, in physiological conditions, and then move on to the pathological ones. So to give you an example of what one can dream of course, you all know, for instance, such projects as a human protein atlas, yeah, where you can nowadays just type certain protein in, and you could see the distribution of those proteins in the different tissues, and also the gender specificity, and so on and so on. So can we dream in the future to have a human lipid atlas where we can also look into these lipid collectives and how they are reflected in the different tissue, in different organs, and perhaps in one day even in the cell, single cell specific manners. That, of course, will help us understand tissue-specific lipid metabolism, and then we could see the effects of lipid plasticities and remodeling. Obviously, having that in quantitative way, we can define some reference values and intervals, which will facilitate tremendously a clinical translation of lipidomics. We can also think about lipid site enrichment analysis. For instance, if you measure certain lipidome, and you can perform this kind of bioinformatic enrichment, you can really assign it to certain cell or tissue types, and of course the systems biology afterwards. So having all that in mind, like I told, we started a project a few years ago where we looked into lipidome remodeling in human obesity. And there is no need to say that among all pandemics we are suffering nowadays as a society, obesity is actually one of those. And the numbers are really kind of striking if you think about that from 1975, the worldwide population of obese individuals actually tripled. Yeah, so, and nowadays, for the European region, reported that over 50% of people are overweight or obese. Obesity comes not just with increase of body weight alone, but it actually comes with a development of many different complications, cardiovascular diseases, type 2 diabetes, certain types of cancer. And there is a massive research going into this area, but we still do not understand a really underlying mechanism, which, for instance, can differentiate these two very crudely defined phenotypes. Yeah, some people are obese in a metabolically healthy way, unfortunately, the lower portion of those, and some of them are metabolically unhealthy. So that means they have insulin resistance, they have adipose tissue hypertrophy, high inflammation, and ectopic lipid accumulation in the other organs. So disease itself is really heterogeneous, and there is, as I mentioned, this metabolically healthy and unhealthy conditions. There are two different wide adipose tissue depots. How good do we know the difference between those? Gender, age, and so on. And with the trend, which is happening now, that we actually have to treat diseases into personalized manner, in the personalized way. So without addressing this heterogeneity in general, it's hardly to imagine that we can move to the personalized treatment at all. So with that, we started this project. And as I mentioned, Mike was the person who actually made the whole wet lab for this project, and that's why I would like to pass now to him, and he would be the first person to explain what exactly did we do here.
Yes, hi. Good morning, everybody. It's 7:30 where I am. I know it's a bit later for you guys. It's my pleasure to today show what kind of analytical workflow we had developed in order to assess these problems that Maria has just talked about in generating a reference lipidome and understanding pathologically or differences in the lipidome induced by pathology. So here we looked at various different kinds of adipose tissue samples. So we got biopsies from subcutaneous and visceral adipose tissue depots from different kinds of sample cohorts. So we had lean patients and we had a cohort of obese patients that we also subdivided into metabolically healthy, insulin sensitive patients and metabolically unhealthy, insulin resistant patients. So when we started off with trying to analyze their lipidome, we found that adipose tissue is actually an extremely hard tissue to work with. It's full with connective tissue, it's cellularly very heterogeneous. Also, on the lipidome perspective, it's quite complicated to work with because here we have a large or up to a huge dynamic range of concentrations of the different lipid species. For example, represented here by the extremely high concentration of the energy storing triglycerides that are orders of magnitude higher concentrated than like signaling lipids, such as sphingolipids or phospholipids here. And so what we did in order to assess this complexity of the lipidome is that we optimized and tailored an analytical workflow specifically to the adipose tissue. So that means that we optimized extractions, we compared and optimized various different fractionation methods to enrich low abundant lipids and deplete high abundant lipids. And then we used liquid chromatography coupled to mass spectrometry to really analyze the whole broadness, I would say, of the lipidome. So we wanted to have an analytical workflow that is not discriminating between high versus low abundant lipids, but we really wanted to see all of them independent of their concentration and structure. So this is the workflow summary that you can see here. I will go into detail to each of these parts. But what you can already see here is that we couple several different kinds of liquid chromatography to several different kinds of mass spectrometry, high resolution mass spectrometry, to really assess the complexity of this lipidome. And then we applied a very rigorous data analysis workflow that was supposed to cover the entire lipidome, and not only cover it, but also cover it with very high confidence. So therefore, we also curated manually our data, and we performed structure retention time relationship analysis to rule out like false identifications. So this enabled us in the end to get a lipid library of the adipose tissue that is really curated in 3 dimensions, whereas we have the accurate mass, the fragment spectrum, and also the retention time as 3 independent ways of analyzing these lipids. So why is confidence important? I know that not everybody is working in the lipidomics field, and it might sometimes seem abstract why we use such like dedicated workflows. Why do we put so much effort in it? And I wanted to exemplify that based on 2 different phenomena that can happen, especially for sphingolipids such as ceramides. So you can see here two different ceramides depicted. You see that they are consisting of different sphingoid bases and different lengths of fatty acids. But when we look at them with mass spectrometry, we find that they are actually completely identical in mass. These are isomeric species. But we know that they arise from different fatty acid acylation to the different amino acids here. So what we can do is to perform a tandem mass spectrometry experiment in which we can really define the fragments that are indicative for the certain ceramide species that we're looking at here. A problem arises when both of these lipids are present at the same time, because then this results in these mixed fragment spectra, which will not allow us to differentiate between these two different species. So when we do the quantification afterwards, we can only quantify them together in bulk, basically. And this only gives us some number of carbons, some number of double bonds in this whole species structure. So it completely prohibits us from understanding metabolic fluxes. Where are fatty acids coming from? Where are they going? So we had to apply here liquid chromatography systems that can actually resolve these isomers. Another thing that we usually observe is something that's called in-source fragmentation. And I wanted to exemplify this on these two ceramide species. The upper one is a deoxyceramide and the lower one is a dihydroceramide. So for people in sphingolipid field, we know they have a very different function and arise also from different pathways. So when we analyze them with mass spectrometry, we get this fragment spectrum here for a deoxyceramide, for example, which is cool, and we can be quite confident about it. But when we do the same for a dihydroceramides, these molecules are notoriously known for being labile, and upon ionization, they can fragment and lose one of the hydroxy groups that you see there. So this results in an ion that is isomeric and identical to an ion of a deoxyceramide. So that would be a false positive identification. And what that leads to is that we would also make a completely false biological elucidation in the end. So we want to make sure to remove these false positive identifications. And therefore, what we do is that we apply liquid chromatography and we try to correlate the structure of each species to its retention time on the liquid chromatography. And this is what we did in this workflow. So first of all, we wanted to separate isomers. Lipids are very broad in structure, so we had to develop and apply several different kinds of liquid chromatography to be actually able to separate isomers from different classes. And we then connected it to mass spectrometry. And as I said, we wanted to be here non-discriminative. So we used untargeted methodologies, conventional data-dependent acquisition, but also deep scan acquisition modes like AcquireX technology. And for the ones that were very low abundant, we also used targeted parallel reaction monitoring approaches. And you can see in the middle the chromatograms arising from these analyses. And you see that with the different kinds of chromatographies, we are also able to separate isomers from different types of lipids. And then on the right-hand side, you can see the retention time mapping that we have performed. X-axis is the retention time and the Y-axis is the Kendrick mass defect, which is a measure for the structure of a lipid. And you can see here if lipids of a similar structural feature follow a linear trend, that means they follow the expected retention time structure relationship. Everything that falls out of it, we exclude it as false positive. So this rigorous identification actually enabled us to be highly confident about our identification processes here. And not only that, but we also curated manually all of these identifications to be extremely confident about their identity. And this is what we came up with. We were able to identify over 1,600 lipids with very high confidence to the molecular species level. And we were even able to differentiate between usually hardly differentiable lipids like plasmalogen phospholipids and ether phospholipids, but also between dihydroceramides, ceramides, and sphingodiene ceramides. Yeah, so this qualitative description of a lipidome also doesn't really tell us much. In order to do some biological elucidation, we have to have quantitative values. We have to know what's the ratio of lipids to one another. What do we actually see here? So I'm not going too much detail into too much detail here, but we have also developed a quantification approach that gives us very high quantitative accuracy. So we use several different lipid class specific standards that were isotopically labeled or not labeled in order to cover the whole range of lipids we had identified from this tissue. We again separated all the different isomers on different liquid chromatography systems and measured in an untargeted way the mass spectrometry here. What we did then is that we again performed the data analysis workflow, ensuring high accuracy. One thing that is quite important here is that we took into account specific adduction behavior and in-source fragmentation behavior. So each lipid can adduct with different kinds of metal ions or protons, and this is dependent on the structure. And you can see that on the lower hand side here. This is on the x-axis, you see various types of triglycerides. Y-axis shows the relative intensity of different adducts to the total ion intensity. And we see that depending on the structure, there's a very different adduction behavior. So we always have to quantify all of these adducts, sum them up together. Additionally, we performed some isotopic corrections, and I also developed some response factors for triglycerides that I'm not going into detail with. But main outcome here is very high accuracy of the quantification, very high confidence of the identification. So we are very sure about what we actually measured here. And so this is what we came out with. This is what came out of this analysis, which is our AdipoAtlas, which is the first quantitative reference lipidome of the human adipose tissue. So we were able to semi-absolutely quantify over 700 distinct individual lipids. And you can see here that each horizontal line is one lipid class, and each vertical line in this lipid class represents a single molecular species. So this is really cool because now we can see that lipids in the adipose tissue span over a huge range of concentrations, over 8 orders of magnitude we can see here. For example, between a very high abundant triglyceride and very low abundant acylcarnitine. And also within a single lipid class, the triglycerides, for example, we can see they range over orders of 5 orders of magnitude. So this is extremely astonishing to us, and I hope also for you. And I would give the stage back to Maria, and she's going to talk to you about some more interesting findings we could get from this analysis.
Thank you, Mike. And so you could see actually there was a lot of work, right, to make this kind of picture. And of course, there is a lot of data behind this picture for all those lipid species with the quantitative values. And the question is, what did it give us afterwards? Yeah, so did we learn something more which we didn't know before? Was it a sense to do that in this very time-consuming, rigorous, deep lipidome profiling? And of course, there is a lot of data in there. I will not be able to show you all of the examples, but just provide you a few of those. And since we are here in the Sphingolipid biology webinars, also beyond Sphingolipids, I still would like to show you some example on the Sphingolipidome of the adipose tissue. So Sphingolipids, of course, everybody in this audience, I guess, know better than me, are a very interesting class with a lot of functional activities assigned. But what we generally know that ceramides are known to induce insulin resistance. And they do that in their, let's say, so they act as a metabolic sensors. So they may sense, let's say, oversupply of free fatty acids, and especially saturated free fatty acids. And when the biological system, so it has too many free fatty acids, they had to get rid of that in any way they can, because they are detergent-like and definitely not good to be present freely in the cell. And in this case, ceramide actually signal by inducing the inhibition of AKT signaling, so insulin signaling in this way. They also inhibit, obviously, uptake of glucose and amino acids. So actually to use this free fatty acid as the main source of the energy, to consume them as much as possible. They promote triacylglyceride synthesis, slow down lipolysis, and also decrease mitochondrial efficiency. So on this point, the first event in the ceramide accumulation can serve rather as adaptive mechanism, but if the system still remains overloaded with the free fatty acid supplies, this accumulation becomes cytotoxic, and this is usually what is described in many cases for ceramides. So what do we know so far about ceramides in human white adipose tissue? Of course, this is a topic which is researched for some years because of the obvious connection between obesity and insulin resistance. And indeed, ceramide accumulation in white adipose tissue is happening in obesity and obesity-associated diseases. And there are few ceramide species which actually addressed in this content or in this perspectives. Yeah, so this is this, let's say, classical sphingosine-based ceramides, which are chelated with, for instance, palmitic acid, or there is also data showing that 24-to-1 fatty acids increase also their content in the ceramides. And that is associated with adverse metabolic outcomes. But in majority of the cases, the, let's say, sphingolipidome of adipose tissue was not addressed in untargeted manner. So most of the people perform this analysis using targeted approaches. And majority of them target up to, let's say, 10, maybe 15 species present there. Are there any other ceramides in the adipose tissue, and can we actually describe some depot specificity? And since we have can obtain a quantitative values, can we in the future define really this threshold between adaptation towards really cytotoxic effects here? So when we looked in our AdipoAtlas data in the sphingolipidome of the human white adipose tissue, that was probably the most astonishing results, because we could see that it's much more diverse than this dozen of ceramides which are classically measured. So overall, we have identified and quantified over 130 species of ceramide lipids. And you can see on this Sankey plot, so that they're represented by different classes. So we have these classical ceramides, we have glycoceramides, at least the ones which we can measure regularly, up to 3 hexoses. But we also have other present, like sphingodienine ceramides. I will talk about that a bit more, and the oxyceramides. And the distribution of sphingoid bases is actually also much wider than one would expect, as well as the different acyl chains are also shown here in the percentage. So to show you some example, what actually happens and in obese adipose tissue versus the lean one, we indeed do have accumulation of ceramide as we expected to see. And many species are rising up. And here we provide a summary for based on the different sphingoid bases. And for instance, this one, yeah, which is the most addressed, indeed increase both in subcutaneous and in visceral white adipose tissues. But also the interesting fact, now having these quantitative values, we can see also the difference between those different depots. So if we look at the basic levels of this kind of ceramides are pretty much different between these two different tissue types. And obese subcutaneous white adipose tissue accumulate ceramides up to the level which are actually characteristic to the lean visceral tissue. And that is also the true for the, for instance, ceramides with the base 16-to-1. We can see exactly the same behavior here. But what was also interesting, that we could see a high enrichment in the human white adipose tissue of this so far relatively mysterious sphingodienine ceramides. So this is the ones which are formed by the introduction of additional double bond into the sphingoid-based structure. And actually the enzyme which is responsible for this reaction was characterized only a few years ago. And those lipids were already detected before in the human blood plasma. But here we could see that they are extremely enriched in the white adipose tissue as well. And if you look here on the quantitative values, yeah, so it's really pretty much in the same orders as a regular ceramide, which everybody is usually looking at. But now when we start to see for the differences between subcutaneous obese and lean and visceral obese and lean, we could see that the fold change, and here we have also the absolute quantities between lean and obese white adipose tissue is much higher for the sphingolipids ceramides in comparison to the classical ones which are measured. So we can really say here that this new, relatively new class of ceramides is enriching white adipose tissue and show most significant increase in obese state. So again, the next example here from the ceramide signature. So we compared also the composition, which was reported in the literature recently, of the different of the ceramide derived from the different sphingoid bases in the human blood plasma, with what we have actually in the white adipose tissue. And we can see that in many cases, they pretty much mirror each other. Yeah, so the classical ceramide base, then we have here sphingolipids 16:1. But here we have also quite high number of the oxyceramides. And at the moment when we were submitting the paper, it actually came another paper out which are confirming the same result, that white adipose tissue, human white adipose tissue, is extremely enriched in comparison to plasma with the oxyceramide. What does it mean? Nobody yet know, but we're looking also on the quantitative values in obese versus lean individuals. And what we saw in our data here, that the oxyceramide, which usually would be enriched in human white adipose tissue, they drop down with the development of obesity. And there are reports that they're actually increasing in the metabolic complications in the human blood plasma. So there is a question opens up, is white adipose tissue serve as a reservoir and maybe even the source of this deoxyceramide, which can be released in the circulation in the certain metabolic distress conditions. So again, with all those data which we have now, we became definitely closer to understanding, or at least formulating some hypotheses what can happen in all this ceramide accumulation line. So again, with accumulation of free fatty acids, they either go to the beta oxidation, they can accumulate in the lipid droplets, but at certain moment it might be just much too much. So the ceramide synthesis increases, and we have accumulation of ceramides, which leads to different action. So there are definitely some adaptive responses and some lipotoxic responses. And I think the key to answer this kind of question, which is this ceramide molecular specificity, and actually also their paracrine or endocrine signaling afterwards. So then the next example, which I would like to show you now, is coming from the other class of lipids, is triacylglyceride, which is, yeah, it's obvious. Adipose tissue is really rich in triacylglyceride. There is nothing new here, and it's the most abundant lipid class. This is something what we expect. But this class is super chemically diverse. Yeah, so we identified alone from triacylglyceride more than 1,000 of molecular species, yeah, combination of different acyl chains. And then we quantified them on the species level, because this is really the composition which is very hard accurately to resolve. But the interesting fact, when we looked in our quantitative data, we could see that actually 70% of the total abundance of triacylglyceride in the human white adipose tissue can be explained by just 20 species present here. Yeah, so the rest, the 1,000 rest, is still there, but they do not make a significant impact. So what we could see here, that there's 20 species which make up to 70% of the total TG amount in the adipocytes, are represented mostly by saturated or monosaturated fatty acyl chains. Then when we look in the obese condition, we start to see an increase in the triacylglycerides. Again, no surprise here, but those ones which are accumulating more in the human white adipose tissue in obese conditions are actually polyunsaturated ones. Yeah, so you could see, so here shown all of them are actually significantly different, but some are having higher or lower changes. So most, for instance, this is the one which contains arachidonic acid. So what happens to these massive lipid droplets in adipocyte if we start to accumulate TGs with polyunsaturated PUFAs? So we thought, of course, first that the presence of polyunsaturated acid chains decrease the packing density. And we ask the following question. So can enrichment of these PUFA TGs drive lipid droplets hypertrophy? And by extension, can it drive adipocyte hypertrophy? And would that enrichment of PUFA TG actually correlate, as hypertrophy usually does, with a certain metabolic complications? So we don't have all answers here, but definitely got more ideas. So we check it out in different ways. So can PUFA TG drive lipid droplets hypertrophy? For that purposes, Mike again actually performed the experiment when he designed artificial lipid droplets, resembling them in vitro. And in these artificial droplets, we can modulate the composition of the phospholipid monolayer, but also the composition of the lipid core, neutral lipid core. And what we could see here, that by increasing proportion of PUFAs in the phospholipids monolayer, lipid droplets become actually smaller. But if we start to increase the proportion of PUFAs acyl chains in the triacylglyceride, in the neutral core, those lipids become larger. So here in this, at least in vitro experiment, we can see this clear correlation that increasing proportion of PUFA acyl chains in TGs drives lipid droplets to become bigger. So the next question is, can a PUFA TG drive adipocyte hypertrophy? So this is something what is still not published. So obviously having this AdipoAtlas data, and we spend a lot of time to dig very, very deep into the lipidome, but now we have this inventory of lipids, and now we can go for very high throughput screening of those lipids which we found in the larger sample cohorts. And this is what we did also. For instance, here we had individual samples of white adipose tissue, subcutaneous or visceral, also some plasma, and we performed the same analysis what we did, but now in much faster way, since everything is identified already and characterized in this 170 samples. So we also had some clinical data or characterization of those adipose tissue deposits, and we looked if we can correlate the mean adipocyte size with the lipid signatures which we see. So, and you could see here in this model that we actually were able to obtain a pretty good correlation. And the lipids which are actually driving this correlation are mostly PUFA TG present here. Yeah? So you could see it here. And another question which we asked, that would this PUFA TG actually correlate with insulin resistance? And I guess it's pretty much already shown here by the colors. So this, but here you can see it as well. So using this lipidomic signature again, we can very nicely differentiate, and this is an example of subcutaneous, insulin sensitive or insulin resistant tissue. So again, lipidomic signature here can differentiate in the supervised analysis those two different phenotypes. And again, the lipids which are responsible, which drive this separation, especially in case of IR samples, are those PUFA TG lipids. So using this all together, we could explain or provide this kind of functional links that the accumulation of PUFA TG in obesity is most probably provides the driving force for hypertrophy of the lipid droplets, hypertrophy of the hepatocytes, and associated complications here. So to conclude this long story of AdipoAtlas, Here we have, of course, accumulated a lot of data, and I provided you just a really few examples. Yeah, so some examples of the ceramide field, but also examples of what we learned here about adipose tissue biology in terms of triacylglycerol roles. But there is much more data there. There are interesting observations for cholesterol esters, for plasmalogen PEs, for instance. Yes. So which turned out to be the fourth most abundant class of lipids in the human watt. Yeah, so usually they are attributed mostly for brain or cardiac tissue, so, but their content is actually higher than the content of sphingomyelins. So also a lot of interesting data on that, cell carencins and so on and so on. The nice thing is that actually those data are all freely available. And if you are interested, you can, if you really like mass spectrometry and want to look in the raw data, you can download raw data from Massive. Or if you don't want to bother with that, you can go also to Metabolomic Workbench, where we uploaded the metadata, but and also the quantitative data. And the guys there in Metabolomic Workbench, they make amazing work because you can actually use their tools, for instance, the statistical tools, to really just question the data sets which are there. Yeah, so you can just go there and make some statistical analysis with our AdipoAtlas there. And the final thing which I would like to actually emphasize, maybe it's not extremely obvious, but it is there. The majority of the study, when we try to understand what happens with the lipid metabolism, it goes this classical way. We formulate hypotheses based on some usually cell culture model results. We use an murine model, for instance, to prove them. And in some cases, it's still coming to the human data validations. But here we go other way around. So the hypotheses which we now formulated here and which I showed here, they are originally deriving from human data. Of course, now we can and we do the improvement of the mechanism using marine models or cell cultures, but I think that increased pretty much our chance to validate those data set finally back to the humans. And having this kind of reference lipidomes, I believe, will help all of us understand finally human biology. So now we have reference lipidomes for visceral and subcutaneous adipose tissue, but that would be, of course, super exciting to have it for other organs and connected between each other. So with that, I would like to conclude and also to thank people who were involved in this work. So it's, of course, Mike who presented today, but it's also a lot of work were done by validating all those lipid identities. That was done a lot by Georgia here and by me. And also, of course, a lot of thanks to Matthias Bluer, who provided us with the samples, and Laura, who helps us a lot with chemoinformatic analysis. And thank you very much for your attention.